Hi everyone, today's lecture is about the multicellular and social characteristics of microorganisms. So you probably haven't thought very much about microbes being multicellular. The one thing most people know about microbes is that they're single-celled, and you probably haven't thought very much about them being social. And so that's the purpose of this lecture, is to introduce you to these concepts, and I have four themes, four examples, four characteristics of explaining my argument for why we perhaps should think about microbes as being multicellular and social. The first one is cellular differentiation, which is being shown in this image. Many, perhaps most, if like nearly all microbial species have some form of ability to differentiate their cells, that is, to have different cell types within one population of cells, with different cell types being adapted and specialized for different functions. So in this image, we have a structure being formed by a microbial species. The little green and red shapes are individual cells. The red cells have committed suicide, a form of apoptosis, if you've heard about that before in a cell biology class. It works completely differently in bacteria than in eukaryotes, so the molecular mechanisms are not the same as those you may have learned about in a cell biology class. But it is analogous in that these cells that are stained in red have intentionally killed themselves in order to help build the structure that enables their sister cells to disperse and carry on the population. So that's one example of cellular differentiation. One of the most dramatic examples is in the cyanobacteria that form these heterocysts. So these are chains of cyanobacterial cells. Each segment in this chain is one cell, so you can still think of it as a single-celled organism if you like, but the single cells are connected to each other and exist in nature as these long filaments, and also big aggregates of filaments. And often these filaments include specialized structures called heterocysts. These are single cells that have differentiated for a particular function, which is nitrogen fixation. So nitrogen fixation is a reaction that is poisoned by oxygen. It can only happen in the absence of oxygen in an anaerobic environment. But these are cyanobacteria. They make oxygen, right? So that is an inherent problem. And their solution to it is to create a differentiated cell where there is no photosynthesis happening, so there's no oxygen being produced. It has a thick cell membrane and cell wall to keep the oxygen out, and then nitrogen fixation can occur inside this heterocyst, and then the nitrogen produced, the nitrogen compounds being produced as a result of nitrogen fixation can then be shared with the rest of the population. Also, there's another example of differentiation in the same figure, which are these spores. So these are reproductive cells. It's a reproductive structure forming spores that will be able to survive difficult environmental conditions. So if all of the rest of the cells are wiped out by a sudden loss of nutrients or a traumatic physical event, like a hurricane comes through and disperses the whole population, then these spores will be able to withstand that dramatic environmental change and carry on the rest of the species. So in this one image, we have three different cell types, at least, belonging to the same species. So an example of cellular differentiation in the bacteria. Some of the most dramatic examples of multicellular structures are produced by the group of bacteria known as actinobacteria. These were originally called actinomyces because they were thought to be fungi, and you can imagine why they were thought to be fungi. Looking at the images of the structures that they produced, these look very fungal, right? But all of these images are actually of actinobacteria. These are bacterial biofilms. A little bit of ecology about actinobacteria. They're extremely known, they're extremely common in soil. They're kind of known as soil bacteria, but they're actually found everywhere. Even in the ocean, there are marine actinobacteria. And they're very, very, very diverse. It's an extremely diverse group of bacteria. They include so-called free-living bacteria, as well as those that live on other organisms, like those that are associated with plants, plant pathogens, symbionts. There are animal pathogens in the actinobacteria. Tuberculosis, for example, is a member of the actinobacteria. The Mycobacterium tuberculosis, the organism that causes tuberculosis. So it's very diverse. The actinobacteria are also the organisms that produce many of our antibiotics. So Streptomyces. The myces is still in the genus name in that case. Again, it was once thought to be fungal. Streptomyces is the genus of actinobacteria that has produced all of the antibiotics that you see here. So really diverse, really useful, really important group of bacteria, and they are their multicellular nature is one of the defining features of this group of bacteria. And here's a cool review about marine actinobacteria and how diverse they are, and the potential for discovering new natural products, including antibiotics and other potential compounds from this group of bacteria that we really haven't even begun to explore yet. More images of actinobacteria, digging a little bit more into the details of their biology. They produce these multicellular structures as part of a life cycle. So perhaps you haven't thought of life cycles in the context of bacteria before, but bacteria also have life cycles. So for example, many actinobacteria have a spore stage. It disperses to a new environment, for example. It creates a mycelium, so it has inherited this terminology from when we thought they were fungi, and because they look like fungal mycelia, but they're bacterial again. The mycelia tend to integrate into a substrate and then produce mycelia that kind of stick up into the air. That's when these spore chains are formed, and then the spores are dispersed in that way. So it's really kind of that's one abstract summary of a general life cycle, and this image is showing the many different forms of these filaments and spore chains that are produced by different species of actinobacteria. So main point of the slide is there is a biology involved here. We actually don't understand it very well, and there's a lot of diversity in that biology, as always. This is that same image from the first slide again. This is actually Pseudomonas rudinosa. It's an opportunistic pathogen. It causes many forms of pneumonia. It's when cystic fibrosis patients get bacterial infections, it's often a Pseudomonas infection. And again, this is an example of the red cells intentionally dying in order to help build the structure that is growing in order to allow the spores, the green cells, to be dispersed more efficiently. And so it's an example of a multicellular structure. Like, this is the purpose of tissues in our own bodies, is multiple cell types are specialized to carry out the function of the larger whole, and that's exactly what's going on here. I don't think we would call this an organ or a tissue exactly, but it is multiple cell types working together for a specific function. Here's a video of what one of these fruiting bodies or spore chains or whatever you want to call them looks like in real time. So this is looking from the side view, and the fruiting body is growing up, and it'll form this tree-like structure at the top, and then the individual spore cells will disperse from the fruiting body at the top. So there's the video, and then a really nice version of that is here. So this is a nicer image of that same fruiting body structure that we saw being formed in the video. And these structures are formed by Myxococcus, which is a genus of bacteria that's really common. It's found everywhere. So it's not some weird alien that's only found in one rock at the bottom of the ocean or something like that. It's an extremely common bacterium that it's probably almost certainly in the room wherever you are right now. In fact, it's everywhere, which is to say that these structures are extremely common. They are a fundamental feature of bacteria. It is not the exception. It is in fact the rule. And the general term that we use for these surface-attached communities of cells are biofilms. So fruiting bodies, I think you could think of as being very specialized biofilms. But in general, biofilms are any kind of community of microbe, could be bacteria or archaea. Archaea also form biofilms that are attached to some kind of surface. So here is a generalized example of a kind of abstract life cycle of a hypothetical organism that forms biofilms. It's in principle the same idea as the spore dispersal life cycle that we saw before. Just showing here to say that it doesn't have to be spores. Any kind of surface-attached community that is a biofilm will have some kind of cycle of individual cells that then attach to a surface and then grow a complex community, and then individual cells will disperse from that community. It's a very fundamental concept to life in general, and microbes are the ones who invented this first, who came up with this idea of forming complex communities, individual cells dispersing, colonizing a new environment, growing another community. So this is a fundamental feature of all life, including microbes. Quorum sensing is a term that has been coined to describe the signaling that goes on among cells during the formation, the early stages of the formation of these biofilms. So when these cells land on a surface, the metabolic and biochemical functions that are required to produce a complex biofilm like this, which means producing all of the chemicals that are extracellular, that are forming like the mucousy, snotty stuff that holds the whole community together and glues them to the surface. All of those functions are triggered by communication among the cells. And they don't want to start doing something that forms a community if they're all by themselves, is the idea here. So these community building functions are triggered when there are enough cells around that would make that those functions make sense. So that's where the word quorum comes from. So when the cells sense that they have a quorum, that there are enough of them that it would make sense to form a community, then that's when these biofilm forming functions are triggered. And the way they do that is that they send signals constantly to each other, and if a given cell is receiving enough signals from its environment that enough of its brothers and sisters are around, then that triggers the biofilm formation. So quorum sensing is the communication that's happening among those cells that leads to the biofilm formation. This is a really fascinating topic. It's a whole research field by itself. The specific molecules that different species use to communicate have been worked out for several examples. So here's a review on this topic, and it's showing how there are already several different examples of quorum sensing. So Pseudomonas and E. coli, for example, have different kinds of quorum sensing. Vibrio has different, the genus of bacteria that includes the species that causes cholera. These all have different systems of quorum sensing, but they all have quorum sensing. So they all communicate so that they know when it's time to form a biofilm. But the chemicals they use, perhaps the chemical languages that they are using to communicate with each other are different, which totally makes sense, right? You would expect different species to have different languages so that they don't get their words confused, because you want to know if the other cells around you belong to the same species or not, right? So this is where the term social microbiology comes into play. The discovery of quorum sensing really opened our eyes to the fact that microbes are constantly communicating with each other. Because again, this is not an exception. This is happening all the time. Almost all species form some kind of biofilm and have some kind of quorum sensing, although in most species we haven't quite discovered how it works yet. Right? So this has opened our eyes to the fact that microbes are really social. They are communicating with each other within a species and also between species. So here's just yet another way of making this point that I really want to get across, that biofilms are the norm. This is the normal mode of life for almost all life in the world. Microbial life, which is most life in the world, is in biofilms. So even in the ocean, which is obviously a big body of water, most of the biological activity, the microbial activity happening in the ocean, is actually happening attached to particles in biofilms. There have been many studies to show that there's more microbial activity happening on the particles in the ocean than happening just with free-living cells dispersed throughout the water column. And so here's one figure just illustrating this concept where all of the activity is actually happening on the particles that are sinking, and you have a whole microbial loop happening associated with these particles as they sink. Sometimes these particles in really productive areas of the ocean can be so abundant that it looks like snow. So this has been described as marine snow when you have so many particles that are sinking through the water. And so here are some researchers who are literally collecting the particles with a net as it sinks because it's so productive. And if you look at these marine snow particles under a microscope, you can see colonies of bacteria in those particles. So again, this is where most of the biological activity is going on in the ocean. Okay, so that was my first example of multicellular and social characteristics of microbes. The second example is gene transfer. So this is perhaps a consequence of the multicellular nature of microbes. We've talked about this before, and I mention it now because it is a fundamental aspect of being multicellular. I've shown this image before that gene transfer is basically a stress response in many, if not most, microbial species. If there are lots of nutrients and everybody's happy, then the cells are well dispersed. There's no reason to clump together. But if you starve them, then the cells clump together, and these different colors are stained or are triggered when genes are transferred between cells in this experiment. So you see lots of gene transfer occurring when the cells clump together, which makes sense because it's a lot easier to exchange genetic material with cells that are right next to you than if they're many microns away, dispersed in the water. So this is a fundamental aspect of being multicellular and being attached to surfaces and living in biofilms, is that gene transfer is going to happen. And so now I want to add one more layer of to this idea of gene transfer from an ecological and evolutionary perspective, which is that genetic exchange can actually increase diversity in a community. And I like to point this out because it's somewhat counterintuitive. You would expect that if there's a lot of gene transfer going on, that the genes in a population among multiple species would be homogenized. If they're well mixed, then everybody is kind of the same. And then if everybody is kind of the same, that means there's lower diversity, right? And that is true in principle, in simulations and theory, if there's tons and tons and tons of transfer, right? If all of the genes are being transferred all the time among all of the species, then yes, everybody is the same. But in reality, that doesn't happen, right? There is a limit to how much gene transfer is going on. So there are models and even experiments to show that with a moderate level of genetic exchange, which is probably most representative of reality, the diversity in a community can actually increase, especially when there are environmental changes or other reasons for a selection event to occur. So this slide is explaining how that would work. To start, we have two distinct species with different sets of genes. Let's say that one gene, this gene A, is shared between species 1 and species 2. So now both of the species share it in this scenario. Now let's say there's a selection event such that only species that have the A gene survive. So in the original scenario where the A gene has not been shared, species 2 is wiped out. It goes extinct, leaving only species 1 because that's the one that has the gene that is selected for by the selection event. But if that gene had been shared, as in this scenario, then the selection event would select for both species, and both species survive. So, really simple example, but shows the principle by which a moderate amount of gene transfer, so one or a few key genes that are necessary for survival or are advantageous in some way, if those are shared among multiple species, and these could be completely unrelated species, right? But perhaps they're unrelated phylogenetically, but they are perhaps related metabolically. So perhaps they are syntrophic partners, which we're going to talk about in a second. Perhaps they're participating at different steps of a metabolic reaction, for example. Then both of these species are survived, and they're and in that way, the diversity of the community is maintained and even perhaps increased as a result of the selection event. So, moderate amount of gene transfer can actually promote diversity and not just make everything the same. Okay, so those were two of my examples of the multicellular and social characteristics of microbes. And let's move on to number 3, which I just mentioned. So this is a good segue, metabolic syntrophy. So I have shown this example before. This is an example of syntrophy. This is an example from our own human digestive system where we eat food that has fiber. That fiber is fermented by a diverse community of bacteria to form small organic molecules and also inorganic gases like hydrogen and CO2. And then there's another level, another step in this overall digestion of the fiber that can include methanogens and sulfate-reducing bacteria. And we've talked about before how the competition for hydrogen and carbon dioxide between the methanogens and sulfate-reducing bacteria will determine whether the gases that you release from your digestive system are flammable or if they're stinky. So if they smell like rotten eggs, it's because you had sulfate-reducing bacteria winning this competition. If your gas is flammable, then it was because the methanogens won the competition. And this is overall an example of syntrophy. The syntrophic relationship, that is the methanogens and or the sulfate-reducing bacteria consuming the waste products of the fermentation. And the definition of syntrophy is a relationship where the consumption of one species' waste products is beneficial and makes the overall process happen faster and more efficiently. So this is an example of that because when the methanogens or the sulfate-reducing bacteria consume the hydrogen, that's a waste product from fermentation. So if that waste product is consumed, that makes the fermentation reaction more thermodynamically favorable, right? It causes the reaction to go more to the right in terms of first-year chemistry. And that means overall more fiber is consumed because the whole thing is more efficient. So that is a syntrophic relationship. Multiple species cooperating to make an overall metabolic reaction happen more efficiently. Here is another example of that syntrophy. In this case, it's ethanol fermentation, which could happen in your gut, but more often this happens industrially. So there's often an industrial need to degrade ethanol and other organic acids, and alcohols and acids. And here I'm just trying to describe in a little bit more detail why syntrophy is important evolutionarily as well as ecologically. It's because it's all about the evolutionary fitness of multiple species being connected to each other. So in this example, we've got an ethanol fermentating bacterium. It's producing hydrogen as its waste product. This is very, very similar to the previous example. Here we have a methanogen. It needs hydrogen. Here's all this hydrogen being consumed by one organism. If the methanogen consumes the hydrogen, then everybody's happy. Okay, so just kind of explaining this concept one more time in a slightly different way of how the methanogen consuming the waste product of the fermentative bacteria not only is good for the methanogen, it's also good for the fermentative bacteria, and it's also good for the overall reaction from ethanol to methane, for example. And this is not something that only happens in our gut or only happening in an industrial bioreactor. This is perhaps one of the most fundamental ways that microbes interact with each other, right? This is extremely representative of how microbes in nature are interacting with each other. And that is to say that microbes are not only competing with each other, there's a great deal of cooperation of syntrophic and symbiotic relationships where multiple species have their evolutionary fitness tied to each other. And so selection events can often select for multiple species at the same time for these reasons. It does not mean that they are permanently tied to each other, right? Evolution is complicated. They may find a different partner. They may evolve the ability where they don't need the syntrophic relationship anymore. But it's a really important way that microbes are organized ecologically and has important evolutionary consequences. One more example of syntrophy that is a really cool example and is good in this class because it allows me to talk about a real ecosystem in nature. And this example is the anaerobic oxidation of methane. So this is occurring in many marine sediments. So here we have the ocean, the seafloor. Underneath the seafloor, we have sediments. And marine chemists had noticed for a long time that the concentration of sulfate in the seafloor goes down according to some trend. So the ocean is very abundant in sulfate, so the sulfate will diffuse down into the sediments, and then it decreases with depth as if something is consuming it, right? Now, methane is produced down below because methane is produced by methanogens. Methanogens are anaerobic. They are often the very last ones on the redox tower chain, right? The electron acceptor from methanogenesis, which is carbon dioxide, is one of the worst electron acceptors. So methane is always being produced very deep down in the sediment column where it's most anoxic. And the concentration will also decreases in many places, not everywhere, but in many places in the world, the concentration decreases as you go up, as if something is consuming it. So marine chemists have been looking at this for decades, like in the middle of the 20th century to late 20th century, and seeing that the sulfate and the methane are often going down to zero at approximately the same point in some of these marine sediment columns. And so the chemists predicted that there must be some microbe that is consuming sulfate and methane. Methane being the electron donor, sulfate being the electron acceptor, and if you put this on the redox tower, indeed it is favorable, but not very much. It's a very short arrow on the redox tower. You would get very little energy from this reaction going from methane to sulfate, because methane is only a so-so electron donor, and sulfate is only a so-so electron acceptor. Usually when sulfate is the electron acceptor, you're using hydrogen or an organic compound, which are much better electron donors. And so the microbe said, no, we've never seen that before. You don't get very much energy out of it. And then even there were calculations that you wouldn't even get a single ATP molecule from this reaction. And so for a long time, there was this disagreement between the chemists and the biologists until finally, around the 2000-ish, when I was a graduate student, it was finally discovered that indeed this reaction occurs biologically, and it's done not by a single species, but by a group of species, by a complex multi-species biofilm. It's possible that a single bacterial species and a single archaeal species cooperating with each other can do this, where the archaea are using methane as electron donor, the sulfate-reducing bacteria are using sulfate as the electron acceptor, and then there's some kind of transfer from the archaea to the bacteria to make this happen, right? So it's a syntrophic relationship. Archaea are converting the methane into something. That something gets transferred to the bacteria, and the bacteria use sulfate as the terminal electron acceptor. But for a long time, it wasn't known what was being transferred. It wasn't known exactly what the archaea were doing or what the bacteria were using from the archaea. And this was a relatively recent discovery, and it probably doesn't even apply to all cases of this relationship, but at least in some cases, we appear to have figured out what's going on here. And in the study, they had three different models. One idea is that hydrogen was being transferred, so this would be the same as the previous syntrophic relationships that I've shown you on these PowerPoint slides. So that was one option. Second option is that some form of sulfur was being transferred between the cells. And the third option is that there were wires, like actual electrical conductive wires transferring electrons between the cells. So instead of some kind of chemical compound, the idea is maybe they were just literally transferring electrons, which means that they had an electrical wire from one cell to the other. And this has been considered for a long time, but until recently there wasn't much evidence for it. And now, in the study here in Nature, they had these amazing images of things that look very, very much like wires, and they did a bunch of experiments to show that they were in fact conducting electricity, that is electrons, between the archaeal cells and the bacterial cells. So here are archaeal cells, they're consuming the methane, they're ripping electrons off of that methane. Those electrons travel by electrical wire to the bacterial cells. The bacterial cells use those electrons to reduce the sulfate to sulfide. And so this, at least in some examples, some species that do this, in some locations of the ocean where this is happening, the solution to this mystery from decades ago is apparently that you can get enough ATP from this reaction if you are so efficient in transferring the electrons that you're actually using wires to shuttle the electrons from one cell to the other. All right, so I think that's pretty cool. I hope you do too. We can talk more about it in class. I want to end with, again, highlighting this point that I'm making over and over again, that this is normal. This is how almost all bacterial and archaeal species exist in the world. They are in some kind of metabolic relationship with other species. So I am using this figure from this great review to show that, for example, in a boring patch of soil, most of the life in that soil is organized as biofilms, and those biofilms have multiple layers of syntrophic relationships where complex organic material is decomposed down into smaller and smaller compounds, and each step of those decomposition processes are carried out by different species, and those species are connected to each other in these complex relationships. So you have these metabolic relationships, you have these social relationships because there's lots of signaling going on, certainly within species, and perhaps even signaling between species. And so this is just the figure kind of summarizing this idea of microbes being both multicellular and social, and that is normal and not just an exception in a few weirdos. Okay, my final point I want to make about why microbes are multicellular and social is really just an excuse for me to show this video that you may have seen me show before in another class of Myxococcus. Myxococcus are predatory bacteria. They eat other bacteria. So here are images showing a colony of Myxococcus invading and killing a colony of E. coli. So if you imagine a Petri plate and the little dots, the little colonies on the Petri plate of E. coli, here is one colony of E. coli, and here is a swarm of Myxococcus swarming over the E. coli colony and eating it. They've been called wolf packs because of how they swarm over their prey and consume them. And also, wolf packs is a good analogy because they are cooperating with each other. So the individual cells of the Myxococcus are actually interacting with each other, presumably exchanging some kind of information and some kind of communication system that is still being researched. And they seem to be exchanging information about where the prey is, because they are able to devote more resources and direct the swarm in the direction of the prey once some of the cells detect it. And so here you can see how the swarm has directed its motion, right? It's not just going symmetrically in all directions. Once it found the E. coli colony, it directed the swarm to the E. coli colony. It is now completely covering the E. coli colony and consuming it, and it produces these black spots. These are fruiting bodies. So the fruiting bodies are producing spores. The fruiting body formation is triggered once the cells in that location realize that there is no more prey to be consumed. So that's why you had a lot of fruiting bodies initially formed over here because there's no prey here. So some subset of the population decided it was more advantageous to produce a fruiting body and produce spores because there was nothing to eat. And then when they went over here and ate all of the E. coli, you saw the production of the fruiting bodies after all of the E. coli was gone because there's nothing left to eat. So you might as well produce a fruiting body, produce spores. Hopefully those spores will disperse in the wind or in the water and then go find some more E. coli somewhere else. So multiple examples of cellular differentiation, multicellular behavior, response to environment, response to another organism, so social and multicellular characteristics. If you want to see a little bit more about how this works, the study included many different videos at different scales, so you can see how the cells are interacting with each other. And this one, it's zooming in a little bit more on these wave-like patterns that we saw in the first video. The wave-like patterns are caused by alternating densities of cells, which occurs because in this next video you can see even better because it's zoomed in and slowed down. The cells actually pause and exchange some kind of information with each other. As you can see in this next video, that is zoomed in and slowed down so that you can see when the cells interact with each other, when they encounter another cell, they stop, or at least slow down, and they seems to be like they're talking to each other. Presumably they're exchanging some kind of bit of information, presumably about where the prey is, and then after that interaction, the cells decide to either both go in the same direction or go into opposite directions. And from that basic interaction, stop, communicate, decide to go in the same direction or decide to go in opposite directions, multiply that by a whole population of millions of cells, and you get this wave-like pattern. The wave-like pattern is from these alternating densities of cells. So a wave of cells that have paused and interacted with each other, a wave and just the space in between where there aren't very many cells, and another wave of cells pausing to interact with each other. That zoomed out causes this rippling wave-like pattern here if you let it go faster and on more zoomed out. And the consequence of that, again, is that they find their prey and eat it. So again, I think these are single cells, right? They each cell is acting as a single cell, so I would not necessarily call this a multicellular organism. I don't think this is a tissue, right? They're not forming a tissue. These single cells are acting independently, but they're interacting with each other to have behavior. Behavior is a response to environmental cues. Their behavior is operating on a multicellular scale. So I've thought a lot about what words to use to describe this, and I think that's how I would say it. I would say these are single cells that have multicellular behavior, or behavior on a multicellular scale. All right, so that's my final example of why microbes are multicellular and social. I hope you enjoyed that, and I'll see you next time.